Nearly every cell in your liver, kidneys, muscles, and heart contains the exact same genetic clockwork as the suprachiasmatic nucleus, the master pacemaker in your brain. But these peripheral tissues share a critical biological limitation. They lack photoreceptors, meaning they cannot independently sense the daily light cycle. Without an external reference, these isolated local clocks gradually drift out of sync. Like a room full of watches ticking at slightly different speeds, the system loses its temporal alignment. If this internal chaos is left uncorrected, the body's metabolic and physiological processes lose their coordination. The suprachiasmatic nucleus, or SCN, functions as a master conductor to prevent this drift. It maintains a unified rhythm, forcing all trillions of cellular clocks to align their timing simultaneously. Managing this alignment requires a systemic network capable of translating light into specific microscopic cellular instructions. We can map this process by examining the specific signal architecture the SCN uses to broadcast its daily reset. The brain utilizes 4 distinct pathways: autonomic nervous wiring, endocrine broadcasts, systemic behavioral cues, and core temperature shifts. To understand how these signals coordinate the body, we must first look at the microscopic machinery they are designed to control. Systemic physiological states are defined by their specific intracellular targets. Inside these peripheral cells, the target is the transcription-translation negative feedback loop, or TTFL. Every morning, 2 activator proteins, clock and BMAL1, bind together to initiate the loop, turning on transcription of period and cryptochrome genes. As P and cry proteins accumulate, they bind together, travel back into the nucleus, and attach to the clock BMAL1 complex. This physical interaction shuts down their own production, completing a cycle that takes roughly 24 hours. Resetting the local clock requires forcing a rapid change in the levels of these core proteins, which shifts the cycle's starting position. Every signal the SCN sends, whether electrical or chemical, ultimately serves to modulate this specific TTFL loop. The SCN communicates through hardwired autonomic nervous system circuits to achieve rapid synchronization. It projects directly to the paraventricular nucleus, which sends sympathetic signals down the spinal cord directly to peripheral organs. Sympathetic nerve endings release norepinephrine, which binds to adrenergic receptors on the target cells. This binding activates the CAMP signaling cascade, an intracellular pathway that communicates directly with the cell's DNA. This cascade stimulates the transcription of PER1 and PER2 genes, instantly bringing the local clock into phase with the SCN. In addition to these neural circuits, the SCN drives a morning pulse of cortisol through the HPA axis. Since nearly all peripheral cells possess glucocorticoid receptors, this hormone surge acts as a system-wide synchronization signal. Cortisol binds to its receptor, which then attaches to specific DNA elements called GREs to initiate PER expression. At night, the absence of light allows the SCN to signal for melatonin release, which tells peripheral tissues to begin their rest cycle. This combination of targeted neural signals and systemic endocrine pulses allows for top-down control over peripheral TTFLs. While the SCN is the master pacemaker, its control can be bypassed by behavioral cues, specifically feeding and fasting. For metabolic organs like the liver and gut, feeding behavior is a more dominant cue than the signals sent by the SCN. Eating causes a rise in intracellular ATP and a shift in the NAD+ to NADH ratio. These metabolic changes activate nutrient-sensing proteins, SIRT1 and AMPK. SIRT1 interacts with clock and BMAL1, modifying them to change how effectively they bind to the BNA. Simultaneously, AMPK phosphorylates and degrades cry proteins, which physically shifts the timing of the feedback loop. This mechanism creates a conflict if food is consumed at unusual times, such as the middle of the night. The nutrient-sensing signals in the liver will override the SCN's hormonal and neural cues, shifting the liver clock to match the food timing, while the brain remains locked to the light cycle. This off-cycle behavior results in a state of internal desynchrony, where metabolic organs are out of step with the master pacemaker. The SCN also utilizes core body temperature as a secondary signal for alignment. A daily fluctuation of merely 1 to 2 degrees Celsius is sufficient to synchronize peripheral tissues. Cells respond to these temperature shifts through proteins called heat shock factor 1 and cold inducible RNA binding protein, or CIRP. CIRP physically binds to the messenger RNA of clock genes, helping to stabilize it. This stabilization ensures that the pace of protein translation in local cells remains tied to the daily temperature shifts of the whole body. The SEN uses these thermal fluctuations to maintain TTFL regulation across various tissues. Circadian entrainment is governed by several overlapping pathways. This matrix maps the synchronization mechanisms, the signals, their primary mediators, and their intracellular targets. The autonomic and endocrine pathways converge on per-transcription to set the clock. In contrast, behavioral inputs like feeding focus on the stability of BMAL1 and cry proteins. For metabolic organs, these nutritional signals can take priority over the neural or hormonal instructions from the brain. This architecture shows that systemic human biology is not a single unified march. The body is a multi-oscillator system, and it is vulnerable to conflicting signals from our environment and our actions. Ultimately, the timing of human behavior is every bit as significant for our molecular health as the light from the sun. Physiological efficiency relies on the alignment between our external behavior and our internal clockwork.